From mushrooms to mold and everything in between, fungi crop up in a variety of forms. It's estimated that as many as 5 million fungal species exist, which means that there are far more species than research papers on fungi. At the moment, there is a world of fungal organisms that haven't yet been identified. Anna Bazzacalupo, a PhD student at the University of British Columbia, is a seasoned researcher who studies in the botany department. Her fascination with fungi has brought her to British Columbia because the West Coast has an optimal climate for fungi. For Anna, the reasons for studying fungi are numerous.
Because we know so little about fungi. Because fungi are ephemeral, most of them are microscopic. So if you get an environmental sample, you may find many fungi that you had no idea were there.
But little is known about this unique class of life. Anna and her team of researchers are using a new technique known as next-generation sequencing, or DNA barcoding, which will allow them to easily identify different fungal species.
DNA barcoding can be seen very much the grocery store when you have different products and you have a type of product and that has a specific barcode, and it works the same way in organisms.
DNA barcoding is a useful tool with broad applications. For instance, some antibiotics are derived from fungi, so knowledge of how a specific fungal species exists in the human body is an important part of medicine. But this is just one of the many possible uses for this technology.
Next-generation sequencing is a very powerful technological tool, especially in the most recent years, because it's able to take any environmental sample, say a little cube of soil, a swab from your cheek, anywhere from your hair, and it's able to recover all the organisms that are there if you ask for the right barcode.
Perhaps most importantly, next-generation sequencing will eventually allow scientists to access a database of barcodes for every species. Identification of unknown organisms and even classification of new species will benefit from this technology. For Anna and other researchers, this means fast and efficient communication of knowledge. Although there is a long way to go to identify all 5 million species of fungi, scientists are well on their way thanks to Anna's work and the applications of DNA barcoding.
It has been important for fungi before because unlike plants or animals, in fungi you're able to recover not just new species but even new lineages. Just because you've never seen them, you're able to pick them up through the DNA.
In other words, there's much more to the fascinating field of fungi than just mushrooms and mold.